Good morning, everyone. Welcome back. Hope you had all a good evening and are fresh and ready for a very exciting day of science. So I will be chairing the current session around regulation, and our first speaker is Ashley Sanders. There we go, from the MDC, BIH, and Charité in Berlin. And Ashley was actually before the MBL, so we know each other for a while. And she'll be talking about measuring somatic mutations and their consequence to identify and target subclones.
Great. Hello. Good morning, everyone. It's so nice to see us together again after a long hiatus, and also to start the second day with you. I'll admit that if you're anything like me, the brain is a little slow to activate in the morning, but I'm hopeful by the end of my talk, at least one of us will be awake. So, in case you missed it, my name's Ashley Sanders. I'm a new junior group leader who just started at the MDC last year. And I was recruited into a single-cell focus area that was formed between the MDC, the BIH, and the Charité. And we have the very modest goal of translating single-cell technologies for personalized medicine to improve patient care. And there's actually a nice group of us here representing the focus area. So do keep an eye out for what's coming out of this really exciting team. So my lab is especially interested in studying genomic instability and somatic mosaicism. And this really involves a paradigm shift, where instead of just considering the genome that we inherit from our parents, so our germline genome, we're now considering the genome of every single cell in our body. And that's because during normal development and aging, new mutations can form in our cells. They can clonally propagate, resulting in many hundreds or even thousands of single-cell genomes emerging over time in each of our tissues. Understanding how these processes or how these mutations evolve is really an emerging field. Now, most studies agree that there's a linear relationship between age and mutation burden, and that's because mutations are forming in all of us all the time, even right now. However, the vast majority of this work has really centered on only studying single nucleotide variants. So these are point mutations that are arising in our genome, and that's really because SNVs are relatively easy to detect from sequence data. However, structural variants are also very common, and these are much larger genomic changes in our DNA that can change the number of copies or also the organization. So they're copy neutral, including inversions and translocations. These are much harder to detect from sequence data, and so they're vastly understudied. But just because we can't see them doesn't mean they're not important. And in fact, what we learned from the cancer field is that over 55% of cancer driver mutations are in fact structural variants. This highlights that SVs are forming in our tissues all the time, and that they're disrupting cell phenotypes to generate these pathogenic subclones that really can drive disease. Understanding how this is contributing to other complex disease types is really a big question in my lab. We want to understand how somatic mutations are forming in our tissues, what cell processes they're disrupting, and when they're contributing to complex disease phenotypes. How we're addressing this is by using a single-cell DNA sequencing technology that I was involved in developing during my PhD in Vancouver, Canada. It's called CanSig. It's not only single-cell, but it's also single-strand. And we do this by removing one strand of DNA in the sequencing protocol. We're actually sequencing only half of the cell. And I hope that I'll be able to convince you how sometimes sequencing half the DNA in a cell is actually more informative than sequencing all of it. So, and this is just a representation of the type of data we get, where we get whole genome sequence data for every single chromosome within the cell. And we are able to do this by using a clever biotechnology or molecular biology technique. So before I thought I'd put this slide in just for the new trainees here who haven't seen CanSig before. And before I tell you how this works, I first want to remind you that DNA is double-stranded. So what that means is that each strand of a DNA molecule can be distinguished based on its 3' to 5' directionality. We have a Watson strand, shown in orange, and a Crick strand, shown in blue. Also, humans are diploid organisms. So that means that we have 2 copies of every chromosome within our cell. One we inherit from our mother, and one we inherit from our father. Most next-generation sequencing technologies completely ignore this information, but StrandSeq specifically takes advantage of it. And this is how. During normal cell division, each strand of a DNA molecule serves as a template for DNA synthesis. We culture cells in the presence of BRDU for one round of DNA synthesis, allowing us to label the newly synthesized DNA. That's represented here by the dotted line. Each of the dotted lines contains BRDU, and the template strands do not contain BRDU. This is really important because it allows us to selectively remove one of the DNA strands during the library preparation. During mitosis, these sister chromatids segregate into daughter cells, and it's at this stage that we capture the cell and we initiate our library preparation. And we do so without any whole genome amplification, which is quite unique for a single-cell DNA sequencing protocol. We ligate on forked Illumina adapters, and then, at this stage, we remove the BRDU strand. This is using a Hoechst UV treatment, which creates photolytic cleavages at the sites of BRDU incorporation, leaving only the template strand available for sequencing. We then sequence and align these data to the reference genome, and depending on whether they align to the minus or the plus orientation, we're able to actually detect which strand the cell inherited. And each strand is actually representing an individual homolog within that cell. So we're so interested in sequencing just template strands because it allows us to do some pretty cool things in genome biology. And I put some references here, some of my favorite papers, but really over the last 10 years, we've been developing many applications for this technology that allows us to study human populations by measuring the genetic variation across a diverse human reference panel. We're also using it to study the mutations that are enriched in human disease populations so that we could better understand how their disease is forming. And of course, we're using this to study somatic mosaicism and cellular heterogeneity. And the stories that I'll tell you today, I hope kind of touch on all three of these different broad applications of the technique. So during my postdoc at the EMBL, I was able to develop a new computational approach for detecting somatic structural variants in StrandSeq data. And this was done with Jan Korbel in Heidelberg. And what we did is we designed a new framework that interrogates 3 distinct data layers that are made available through strand-specific sequencing to detect a diverse spectrum of somatic SVs in individual cells. And what we're measuring include the depth of sequence, so that's the total number of reads. The resolution isn't the best. The color wasn't that preserved, so I hope you can see it. Here's the depth signal, where we're just really counting the reads in bin data. We also have the strands, so the number of reads that are present in the minus versus the plus orientation, and also the phase. And we get that because we know that each of the individual strands are representing each homolog in the cell. So we're immediately able to haplotype phase these data. For a reference state, where we don't have any structural variant, we would have a depth of coverage of N of 2. We can have both strands being equally represented, and of course we would have both parental homologs present at the location. This is distinct from a deletion event, which causes a reduction in the read depth signal. We only have one strand being represented, and we have a complete loss of the haplotype that contains the deletion. So we're not only able to identify the SV, but we're immediately able to phase it. Now, these deletions are relatively easier to find from sequence data. Where we really shine is detecting these copy-neutral rearrangements, such as inversions. And this became the focus of my PhD, in fact, where the depth signal doesn't show any reduction because, again, it's a copy-neutral event. Instead, what we see is a signal in the strand where now, because of the inversion, it flips the orientation of a segment of DNA within our genome. We also have a flip in the strand orientation, allowing us to immediately identify the inversion, which would be invisible in other sequencing data. And once again, the haplotypes that contain that inversion are also flipped, so we can, again, immediately phase that inversion. So using these 3 data channels, we've described diagnostic footprints for several disease-relevant SV classes. These include your copy number changes, deletions and duplications, your copy neutral changes, inversions and translocations, and also a variety of more complex rearrangement processes like breakage fusion bridges and chromothripsis events, which I'll describe later. And I just want to drive home the point that the vast majority of these SV classes are invisible using any other technique. And I'm not just talking about single cell techniques, actually. They are also very difficult to find from sequencing thousands or even millions of cells. So we're really able to now dissect more SV classes with better breadth and precision than previously possible. We can do that because we developed a Bayesian model that allows us to systematically test each of those diagnostic footprints from our StrandSeq data in order to predict the most probable SV class at any genomic position within a population of cells. And this is, of course, published work, so I just want to highlight here how we're able to use this to explore the subcellular structure of patient samples and really make predictions about what SVs are present in our patient cells. Now, being able to detect SVs or mutations is just one part of the problem. We also want to know what they're doing, of course. And in order to address that, we've recently learned how to harness MNA's digestion footprints that are made available from StrandSeq so that we can predict the functional consequences of SVs that we are identifying in the exact same cell. And now this is possible because StrandSeq uses MNAs to digest the DNA. And as we know, MNAs cuts between nucleosomes. Nucleosomes is how we package our DNA. And so what we can see at the top right here is that the StrandSeq profile or read density really mimics that's what's shown from an MNA-seq bulk experiment. We're seeing this periodicity in read depth, which is reflecting of where the nucleosomes are being positioned on the chromosome. And in fact, we see quite good correlation between known DNA's hypersensitivity sites and StrandSeq read depth, really showing that the StrandSeq coverage is reflective of MNAs or nucleosome positioning. Now, during gene expression, nucleosomes become depleted from the expressed gene. As a result, you have more MNAs cutting, and therefore less read density at the expressed genes. As expected, we see a negative correlation between gene expression and StrandSeq coverage, where your highly expressed genes, shown in red, show much lower coverage than your lower expressed genes, shown in blue. And we see this not only at the transcription start and end sites, but across the gene body. What this means is that we can use this information to predict, not sure what happened there, oh good. We can use this information to make predictions about what genes are expressed in the cell, which we can use to do cell typing, so identifying which cell types that we have in our StrandSeq data set, and also to make predictions about what genes are on or off within our clustered cells. And I'll give you some nice examples of that in a moment. But I want to summarize this first part, where through strand-specific sequencing, we now have a technology that allows us to not only detect the genome, so measuring the structural variants and other somatic mutations that are present in a patient sample, but we can immediately, from the same assay, detect the epigenetic profiling of that same cell to make predictions about what genes are on and off and correlate that to the structural variation profile or the somatic mutations that we're identifying. And so now we have a tool that allows us to detect mutations in every single cell and predict pathways in each clone that we identify. This is very powerful for understanding how somatic mutations are forming in our cells. Now, being able to identify somatic mutations and functionally characterize them is giving us new information about patient biology. And as an example of this, I want to show you what we found for a CLL patient. So this patient has chronic lymphoblastic leukemia, and they actually were cytogenetically normal. So all the cytogenetic karyotyping performed for this patient wasn't able to identify any genomic abnormalities. However, we sequenced only 75 single cells from this patient, and we identified 11 distinct karyotypes, so abundant somatic mutations forming within this patient sample, which were below the level of detection from other patients or from other technologies. And here I'm highlighting 3 distinct subclones that we identified because we found many of these mutations were actually enriched on the 10q arm of this patient. And at the very top cell, which is a normal cell, we don't see any change in the read density of the strand-seq library because the cell doesn't have an SV. In the next line, we're actually seeing a deletion around 100 megabases, and this is about a 5 megabase deletion. The next cell representing, or the next line representing another clone, shows a terminal deletion from about 100 megabase all the way to the end. And then the third cell here actually has 2 deletions, one at 100 megabases and one just upstream. And so this is really just highlighting the genomic instability that we're able to detect even in a single patient genome. And in order to understand how this is occurring, we really mapped the breakpoints of these rearrangements and found that in many different cells, they were all coinciding with the 10q24 region within chromosome 10. So this is actually a region that's already been associated with poor prognosis for CLL patients, suggesting that rearrangements at this genomic position is actually quite detrimental to the patient. However, we still haven't had, or previous studies haven't had the opportunity to really understand the functional consequences of mutations and deletions at this locus. So of course, we applied our SC-NOVA analysis and identified several disrupted and dysregulated gene networks that were really associated specifically to the 10q deletion. And that's really shown here at this bar plot on the very left, where we have the cells arranged based on whether or not they have a reference allele at the 10q locus, or they have one of those deletions. And we can see that there's quite a shared program of dysregulated genes that are associated with this deletion phenotype. However, when we did a clone-by-clone analysis, we actually found that there was a lot of diversity in which pathways were being activated. If we looked at all of the cells that had any deletion at that position, we saw that Wnt was overall overactivated. We had many Wnt target proteins that were upregulated. However, when we did a cell-by-cell or clone-by-clone analysis, by dividing the cells based on these minor differences in their structural variation profile, we found that there was vast differences in how each of the clones activated Wnt programs. So this, I think, really highlights the importance of being able to disentangle even small changes in the genome in patient samples to really understand how these structural variants are disrupting the different signaling pathways that are important for driving this patient's cancer. So next thing we wanted to find, because we were quite actually surprised to see such diversity in these subclones, despite them having fairly similar SV profiles. We next wanted to validate this using 10x in order to really support whether or not we're able to functionally characterize these cells accurately. So we initiated a site-seq experiment and generated over almost 4,000 single-cell genomes, RNA transcriptomes for this patient sample. And then we applied all of the CNV detection methods that we can find in order to see if we can recover the SVs that we already knew were in this patient sample. And what I really want to stress here is that no matter what we did, we were not able to discover these CNVs. We knew that they were in the patient data. We validated them using every technique that we could find, and we still couldn't find them in the single-cell RNA-seq. Just to really highlight that, here is a zoom-in of chromosome 10, where I just showed you there were multiple SVs and deletions, megabase scale deletions, present within this patient sample. And despite our vast effort, we really weren't able to identify or discover the chromosome 10 deletions within our RNA-seq data. However, when we performed a genotyping method, so we input the SV breakpoints that we already were able to detect or characterize using strand-seq, it was only at this point that we were able to recover some of our subclones. And in fact, we were only able to recover that subclone that had the terminal deletion on 10q. So this deletion was approximately 25 megabases in size. This is a huge deletion. This is almost an entire chromosome arm that's lost in this patient sample, and it's not detectable in discovery mode. It was only recoverable after we knew where to look for it. So I think the main point here is that if you want to know the somatic mutations that are present in your patient samples, single-cell RNA-seq is not the way to get there. You really need some complementary methods to first understand where to look. Okay, so next, we now wanted to ask, okay, we're able to identify somatic subclones, we're able to functionally characterize them, and now I want to give you an example of why this matters. And here, I'll walk you through an example of a T-cell leukemia patient sample, which we actually identified a low-frequency chromothripsis event in. Chromothripsis occurs when you have a shattering of a chromosome that becomes glued back together in a new rearrangement, and is actually very dangerous for a cancer patient. And what we found in this TLL's patient sample is that we had a low-frequency chromothripsis event emerging on the Q arm of chromosome 6. It's low frequency, it's only present in about 30% of the patient's cells, and it emerged at relapse. We know it's chromothripsis because we identified 13 distinct breakpoints that all resided on the haplotype 2 of this chromosome, and that together represented multiple deletions, duplications, inversions, and an inverted duplication. We went on and did 600x sequencing of this patient, and then confirmed that this was indeed a chromothripsis event. So having identified this low-frequency chromothripsis, we then wanted to understand what the functional consequences were. Of course, we performed our nucleosome occupancy profiling, and from this, once again, we're subdividing the cells based on whether or not they have a reference state or the chromothripsis state. We identified a majority or several dysregulated pathways that actually all seem to converge on the MIB transcription factor pathway analysis. So if we did a transcription factor target enrichment, we found that the vast majority of the dysregulated pathways or genes in the chromothriptic cells were all converging really on the MIB transcription factor. So this is indicating that MIB is actually driving the dysregulation of these pathways within the chromothriptic cells. And then we wanted to more functionally or deeply characterize these subclones. So what we tried to do was actually impute the structural variants that we identified in this patient sample, and then identify them in the 10x data, similar to what I just described for the CLL patient, where we used not only the genetic breakpoints that we identified in our SV subclones or in our cancer subclones, but we also use the genetic signature that we were able to identify for these chromothriptic cells based on the nucleosome occupancy profiles. And so we generated a 10x experiment for this patient sample. Again, they had, it was a relapsed TLL patient. And then we used both the conics map to genotype the individual sequencing data or the transcriptomic data, and also UCell to do a gene set scoring of all of the cells based on the nucleosome occupancy profile that we defined for the chromothriptic cells. What I can say is that we learned through this experiment that really it was the gene set scoring that gave us much more power in identifying where our cells were in the transcriptional space of the RNA-seq data. And that's because when we used nucleosome occupancy profiling, we were better able to detect a signature that we can then locate within the expression space. This allowed us to identify our chromothriptic cells in the UMAP, which we found were enriched in clusters 3 and clusters 7. Now that we identified where our cells were within the expression space, we can do more deep profiling of the chromothriptic population. And indeed, what we found is that MIB, the transcription factor, was indeed upregulated in chromothriptic cells, which confirmed our findings from SC-NOVA. And we also did a pseudotime analysis and found that both clusters 3 and 7 were actually much more de-differentiated or early stage in the differentiation of T-cell compartment. So they are primitive cells that highly express MIB and other proliferative transcription programs, really suggesting that these chromothriptic cells are aggressive and that they are prognostically relevant. So having now deeply characterized this small subclone that we identified in this TLL patient sample, we next sought to see if we can learn how to target these cells, specifically based on the information we learned from all of this profiling. And so we interrogated the genes and programs that were upregulated in the chromothriptic cells and identified downstream targets of MIB, specifically Notch1. Notch1 has current drugs that are available which inhibit this pathway. So we decided to initiate an experiment where we took the TLL patient cells and we cultured them in the presence of a Notch1 inhibitor, which is CB103, for 8 or 24 hours, and then we measured the frequency of the chromothriptic clone after treatment with this inhibitor. And indeed, what we found was selective and specific inhibition of the chromothriptic cells. And this, I think, is a really important message. By using the nucleosome occupancy profile to understand which pathways were upregulated, we were able to inhibit these cells specifically. That's shown here, where in the purple line of cells treated with just the vehicle, we have an increase in the 24 hours of this chromothriptic clone. And that's because this clone is aggressive. I've just shown you lots of data supporting that phenotype. However, when we culture the cells in the presence of a Notch inhibitor, shown in the blue line, we actually see a slight reduction in the chromothriptic subclone. And that's because these cells are being specifically inhibited. And in fact, if we look at the expression of Notch downstream targets, we see that in the reference cells, we don't see any significant change in the expression of these targets. We really only see a slight reduction of Notch targets in the chromothriptic cells after 24 hours of targeting. And this also results in the upregulation of DNA damage and stress responses in the chromothriptic cells specifically. So this really highlights how we can go from a single patient sample, characterize the somatic subclones, understand which pathways are activated, and then specifically target those subclones using the information we learned just from one experiment. So I think altogether I've shown you how we really have a very nice multi-omic framework to comprehensively study somatic mutations in patient samples. Now, in this last slide, I just want to really highlight how we want to use this to understand other diseases, not just cancer. And this is because there are many complex diseases that can involve somatic mutations. And so we're using these tools now to measure tissue-specific mutations in the context of distinct diseases, especially inflammatory diseases, to ask what mutations are arising in each tissue, what gene and expression programs are being disrupted, and whether or not we can use this to specifically target or understand how these cells will respond to different experimental manipulations. And we really hope that this will help us be able to understand how somatic mutations are forming in our tissues, when they're problematic, and whether or not we can use this to better treat our patients. So with that, I thank you very much for your attention, and I'm very happy to acknowledge all the people that have worked on this project so far, and also we are actively recruiting postdocs. So if you're interested in some of what I showed today, please get in touch. Thank you very much.
Thanks, Ashley. Indeed, it was a great way to wake up today for all of us. So are there questions? I think, yeah, thank you.
Thanks, Ashley. That's really nice. It's cool that you also had a sort of outlook towards other diseases. I'm wondering how you would be using time points to potentially also sort of reconstruct something like clonal development. And you seem to be having a very good grasp of clone identification, but that part.
Yeah, this is definitely something that we're very interested in. So one, we want to use, of course, the genetic mutations to look at how they clonally expand based on frequencies and what's shared between them. We're also really interested in harnessing some mitochondrial somatic mutations to better, yeah, exactly, with Leif Ludwig, who's one of our great colleagues in the focus area, to really get better phylogenies of these trees. And the idea is...
You can generate time series, or you have some of these already with sort of staged transig or something like that?
I mean, this would be the ideal experiment, certainly. Right now, we're just taking one time point and a late time point for at least one of these studies that we're interested in inflammatory bowel disease. But the idea is that if that yields some really important results, then we would go back and sample the same patients to really see what's happening over time and whether or not these clones are expanding. Yeah, thanks, Fabian.
So we can take one more short question.
Very nice talk. I was actually wondering, you nicely showed that you cannot detect a structural variance using 10x data, but I was wondering, can you detect the consequences of these structural variants using the transcriptomics data or CiteSeq?
Yeah, so in the end, I mean, it's difficult. If you can't find the subclone, how do you measure the consequences? So in the end, we were able to identify at least a very large subclone and then deeply phenotype that subclone based on the transcriptional data. Mostly, it just agreed with what we found in SCNOVA. I don't think it added a ton of information. We learned a lot more from the chromothryptic clone, I think, actually. So the big challenge is, of course, identifying them in the 10x. But once you know where to look, that really, really helps. If you're just trying to discover the SV, you're really underpowered, and you're probably going to miss the subclonality in that patient sample. So really, I think you need a paired approach to study these events.
Great. Thanks again, Ashley, and I'm sure she can answer questions in the coffee break. So thanks so much. So, pointer for them. Pointer for them. So our next speaker, who gets a new pointer, is Stein Aerts from Leuven, and Stein will talk about decoding gene regulation using single-cell multi-omics deep learning and synthetic biology.
Thank you so much. Thank you to the organizers for making this all happen. It's really a fantastic meeting, I think. I'm going to talk about computational strategies that use single-cell data to see, wow. Can I also swap this to the, well, I'll use the spotlight then. It's okay, yeah. So computational strategies that use single-cell data to infer key transcription factor combinations and gene regulatory networks that underlie this cell identity. And we're going to focus on the enhancers where these transcription factors are binding. And for that, make use of single-cell ATAC-seq and enhancer modeling to this way infer enhancer gene regulatory networks. And in the talk, I will start with previously published data on the fly brain, where we integrated single-cell RNA-seq and single-cell ATAC-seq, and then discuss two more recent studies where we took the deep learning models of the enhancers on the fly brain a bit further to generate synthetic enhancers. And finally, I will present a Scenic Plus package. So the fly brain has thousands of cell types, and each of these neuronal cell types have, like in any other animal brain, a unique morphology, unique connectivity patterns, unique function, such as here the Kenyan cells in the mushroom body, T45 neurons in the optic lobe, olfactory projection neurons. And a few years ago, we profiled the single-cell RNA-seq of this whole fly brain. And what now for you probably is not so surprising anymore, but back then we thought it was a really cool finding, is that all these cell types also have a unique transcriptome. And then we set out to do single-cell ATAC-seq on the whole brain. We profiled not only the adult, but also different developmental stages. I will talk a bit less about that. And on this sparse single-cell ATAC data, we applied topic modeling using a package, SysTopic, that Carmen developed in the lab, to optimize topics that are basically probability distributions over both the regions as well as the cells. And then with those topics and probability scores over the cells, you can cluster cells or draw a UMAP like here, showing the development. Does this work? The development from neurogenesis over different stages of pupil development to the adult cell types. Now, if we take the adult cell types, the adult cells only, and here in this case in a T-SNE, we find many distinct clusters. here, and many of them could be mapped and annotated using the single-cell RNA-seq data. For that, we use a gene activity score, and there's really a lot of clusters that have a one-to-one mapping between the attack and RNA. And so now we have here the labels. In blue, the subtypes of the glial cells. In green, all the subtypes of the optic lobe neurons. 3 in yellow, subtypes of Kenyan cells. And then in brown, red, are still many unannotated cell types of the central brain, which are also not yet annotated in the single-cell RNA-seq data. So now we have all these cell types, and for each cell type here in the rows in this plot, there are 20 to 30,000 attack peaks per cell type, and about, no, it doesn't always work. And in the dark color here, there's a few thousand of these peaks that are differentially accessible in that cell type, so we call them DARs. We could also validate this by FACS sorting specific populations of the brain, here Kenyan cells, and found that the peaks that are specific for a cell type in the single-cell attack are also specific in the FACS-sorted cells in the bulk attack. So now we have these DARs and topics. We can try to find motifs and transcription factors and enhancers from these co-accessible regions. We do this in 2 ways. One, by conventional motif discovery, although here we found it important to use a motif discovery method that not only uses a large collection of position weight matrices, but also allows you to calculate a leading edge, so that we can create a critical, or calculate a critical subset of the co-accessible regions that score significantly high for this motif. Then we have candidate motifs. Now we go to the single-cell RNA-seq data to compare this with the expression of transcription factors that can recognize this motif. For example, for ILS, you see that in the x-axis, ILS expression is high in 5 cell types in the brain, and the y-axis, this motif is enriched in the accessible regions of those cell types as well. And the 3 highest ones are the 3 subtypes of Kenyan cells, and indeed, ILS has a severe phenotype in the mushroom body, besides its eye phenotype. We did this then for all the cell types, and then we found more than 100 transcription factors that are both, for which both the motif is enriched in the accessible regions, that's the size of the ball in this dot plot, as well as the transcription factor is expressed in that cell type, that's the red color. So this agrees quite well. It's not easy to validate this, of course, because it's hard to get in vivo transcription factor binding in the fly brain in a specific cell type. We've been trying cut and tag for ILS. It works, but it's not really a super sensitive technique, so the peaks that it finds are good, finds the same motif as we find in the attack. Targeted DAM-ID worked a bit better for ACJ6, also finds exactly the same motif by DAM-ID as we find in the attack data, and the regions also agree. And the second approach we used to further scrutinize these DARs is to train a convolutional neural network. This was work by Ibrahim. So here, the network is trained to, with an input, the sequence, and to predict the topic, basically. So that means where this region is accessible, in which cell type. We did this on a subset of the data with clear cell types, the Kenyan cells, the optic lobe neurons, and the glial cells. And then after validating the model, we can use it to learn something about it. So the first thing we looked at are the features that are used by the model to predict where this region is accessible. This was done using TF-Modisco, and you see that the features that are important for optic lobe neurons are forkhead, ACJ6, and TFAP. Forkhead motif is important in these subtypes of, in these subtypes of T neurons, and that's also where this factor is expressed, and not in T1 and T2, and that's also where the feature is not important. ACJ6 is found for all except T1. It's also the one where it's not expressed. And TFAP is found in all except T3 and T2, and it's also not where it's expressed. So there's an agreement between when this feature is used for accessibility and when the factor is expressed. For Kenyan cells, we find one-cut ILS and MEF2. MEF2 is a little bit less important for alpha prime, beta prime. It's also a little bit less expressed there. And we also find motifs that are negatively important. So this MAMO motif, when that is present in an enhancer, then this region is not accessible in gamma and in alpha prime, beta prime. And it's also the cell types where this factor is expressed. So this could point to a repressor that makes this region no longer accessible for TM5. The second thing we can do with the network is to interpret individual enhancers. Here's the first one is on the T4 neurons, showing the different motifs for transcription factors, and the bottom one is for Kenyan cells with ILS and MEF2. Then we cloned these regions and made transgenic flies, linking them with the minimal promoter to GFP. And indeed, we found that the upper one is specifically active in T4 neurons, and the bottom one is specifically active in Kenyan cells. Together with Tony Southall, we performed more targeted DAM-ID experiments, because from this type of deep explainer plots, we can extract all the TF binding sites, predicted binding sites, over the whole genome, then center them, and indeed, the targeted DAM-ID shows that these are the sites where the factor is bound, ACJ6 and also MEF2. These sites, genome-wide, are also highly conserved. And when we knock down the activators like MEF2, we find that the motifs lose accessibility, one cut as well. And for MAMO, the motifs are gaining accessibility if we knock it down. So this confirms that this could be involved in keeping this region closed. And the third thing we can do is to use saturation mutagenesis to find individual positions in the enhancer that strongly affect the enhancer activity. So here are some examples of mutations that are predicted to abolish the enhancer activity, and when we in fact create those mutations and make new transgenic flies, indeed the enhancer activity is gone. In the bottom one, there are mutations that are predicted to increase the enhancer activity. These mutations mutate the MAMO binding site, and here we indeed find an increase of cells where the enhancer is active. Using this model, we then did a larger screen for Kenyan cell enhancers and found that with a high success rate, we can find Kenyan cell enhancers. So there's all of these here show this patch of green that corresponds to where the cell bodies are here in these two triangles, where the cell bodies are of the Kenyan cell. That's where the enhancers are active. Now I'm going to go back to the global data set after this enhancer modeling. We have transcription factors, motifs, and enhancers. What are the target genes that are regulated? For this, we're linking candidate regions to candidate target genes using a random forest regression with GRM boost. Basically, we are trying to predict from the changes in accessibility what the changes in expression are, and that allows us to go very far in the locus, very far distal from the gene, to link regions to target genes. We also do the random forest regression on the transcription factor to target genes. This is basically the first step of our scenic method, to fish candidate target genes based on co-expression with the TF. And if these target genes agree with the ones that we fish out via the regions, so these two candidate target sets agree, then we have a hit, and we use it to build the GRN. If you combine multiple TFs like that, you have the transcription factors in the middle here, and then the blue nodes here are the enhancers, where they are predicted to bind, and the red nodes are the target genes, and you see that many genes have multiple regions that are predicted to regulate them. Here's another example. This region here is accessible in two cell types, alpha, beta, and gamma, but not alpha prime, beta prime. It's linked to this SNPF gene. This gene is expressed in exactly those two cell types and not in the third. If you clone the enhancer and make a transgenic fly, it is active in the Kenyan cells. And if you give it to the deep explainer, then it tells you where possibly the transcription factor binding sites are. And we did this for 40 cell types, and all these networks are available via the Index website. You can download them and visualize them in Cytoscape. From the brain, we're currently trying to expand that to the whole fly. So within the Fly Cell Atlas community, we recently published the single-cell RNA-seq atlas of the entire adult fly. And in this paper, we have shown that, in fact, the cell types can be very well recapitulated by doing single nuclei extraction from the whole body or the whole head. And this agrees very well with the cell types that we find in the dissected tissues. So there's 15 dissected tissues here on the left, and they integrate very well. This means that, in fact, we can do single-cell analysis on the whole animal, and we are doing that at the moment for single-cell ATAC-seq. And you can see 600,000 cells that we have so far, and now we're trying to port the annotations, and that's quite a tedious task, the labels from the RNA to the ATAC using some multi-ohm bridging in the middle from Satija's lab. For the next part, I'm going to go to the enhancer design. So we have this deep learning model, which gives us nice interpretations. If these are really correct, if these represent really what the Kenyan cell enhancers, for example, are, then can we use this deep learning model to actually create de novo enhancers from scratch? So Ibrahim in the lab devised 3 strategies for that. In the first one, we use sequence evolution. So we start from a random sequence, do saturation mutagenesis, give all these sequences to the deep learning model, score it for a certain cell type or topic, and select the mutation that gives the highest increase towards that cell type. And then iterate over that, do it again and again and again, to evolve the enhancer to a cell type. The second one is motif embedding. So we use a transcription factor motif, implant it at every possible position, score all these sequences with the model, and implant it in the best position. And the third is a generative adversarial network, or GAN, that is trained, and then the generator, after training, spits out enhancer sequences that can no longer be distinguished from real enhancers. Then we synthesize these sequences and clone them with a minimal promoter in a reporter vector, and then for the fly, in vivo work, we make a transgenic fly. For human enhancers, we use cell lines with the transfection. So first, the in silico evolution, we started with 6,000 random sequences and let them evolve. And then what you can see there on the left plot is that already after 10 mutations, we achieve a probability that this sequence is an enhancer that is even higher than the genomic enhancers, only 10 mutations. In the middle plot, you can see that the distance between the furthest mutations is on average 200, so we allow 500 base pairs to change, but most of the events occur within 200, so the space where all the information is contained is usually smaller than 500. And here you can see an example of a random sequence on top, and then the result after only 10 mutations, which is really a beautiful enhancer with ILS, 1-cut, and MEF2 binding sites. And the individual positions of mutations are indicated there. What mostly happens in these mutations, because we've been able to track them, of course, is that the repressor sites are removed and activator sites are generated. And that's more or less what we see happen, not so much in the flanking sequences. For the 6 enhancers that we made transgenic flies of, 4 were positive, 2 were not. We have not found why yet. They look perfect, but only 4 worked. Well, for us that was good. That's already nice. We can do a lot of more fun things with this. For example, we found many enhancers in the fly genome that are actually active in 2 cell types or in more cell types. Like on the right, this enhancer is active in T2 neurons, T1 neurons, and in gamma Kenyan cells. And it's not because they use the same factors, actually they use different transcription factors. Still, it's the same sequence that's active in multiple places. Indeed, the deep explainer in these 3 rows shows you different important motifs for the different cell types. So we reasoned that we could now do the evolution, maintaining the probability for the Kenyan cells and destroying the function of the T neurons. So after evolving that, after 9 mutations, now the Kenyan cell code is still there, the T neuron code is gone, and if we make a transgenic fly, indeed, the GFP is now gone in the optic lobe, and it has remained in the Kenyan cells. The reciprocal of that is also possible. I think this is even cooler. Can we add a cell type to an enhancer? So this is an enhancer that only is accessible in T neurons and also active in T neurons, and now we evolve it to maintain that code and to add mutations that create the Kenyan cell code also inside that sequence. That's happening here. So you can see after the mutations, the T code is still there, and the Kenyan cell code has appeared. And then we make a transgenic fly, and indeed, it's now also active in the Kenyan cells. The second strategy is motif embedding. Here we can also do many sequences, embed motifs, and then afterwards, of course, we have a large collection of sequences that we can analyze. And we found that if you add a MEF2 and an ILS motif, you get an increase in the Kenyan cell probability. But if you add both, then the increase is very, very high. So that's here, M and E. Can I point? This MEF2 and ILS, and then here you go up really hard if you add both. So this shows really cooperativity. And then you can increase further by adding 1-cut on top, but not so much anymore. But still, there's an increase. Also, by analyzing the positions of these motifs that you implant, we found that there are certain preferred spaces between MEF2 and ILS of 5 base pairs, 1-cut and ILS of 3 base pairs. And we reasoned with that, we could actually squeeze the information into a very short sequence, maybe a minimal enhancer, with only ILS, MEF2 and 1-cut, with these optimal positions, and nicely, that's also worked. So here we have a minimal enhancer with 49 base pairs. And I think it's quite profound because it shows that maybe it's not always true, so probably not, but some enhancers can be created only with the motifs. No flanking sequence or nothing needed here. For the GANs, this also worked to some extent. 2 of the 5 that we tested were positive, 1 we are still doubting, and 2 are negative. The GAN does create nice sequences, they look really good, and it also allows to generate the background, so we have a higher GC content in the middle of the enhancer in real genomic enhancers, and also the GAN has learned that, while, for example, a 5th order Markov model would never be able to generate that. I'm not going to go into detail of this, but you can look at the preprint. So we did the same strategies for human enhancers that are active in a certain melanoma cell state, and they also worked here. We could test it with luciferase assays. So we have here 7 out of the 10 in green that give an activity as high as the genomic enhancers and are also specific, and 3 of them do not work. We probably need a larger amount of enhancers to test before we can figure out what's happening with the negative ones, which is, of course, interesting too. Then for the last part, I wanted to go over the Scenic Plus package. This was developed by Carmen and Seppe in the lab, and Scenic was published in 2017 already, a while ago, by Sarah. So what we do in Scenic is to combine co-expression between transcription factor and target genes on the left with motif discovery to prune this network into direct target genes, but here we have to do motif discovery in a very large sequence space, right? We have to look at the sequence around all these genes, introns, upstream, downstream, everywhere. As I've shown in the fly brain, of course, now with single cell attack, we have a much more narrow and precise subsequence space to do the motif discovery in. So Scenic consists of a number of steps, and basically it recapitulates what I've shown you on the fly brain, but we made a lot of improvements of all these steps last year. So we begin with doing topic modeling on the single cell attack data. That's usually the starting point. Here, Carmen developed a new Python package now for systopic, PySystopic, that uses a different implementation mallet for the latent Dirichlet allocation, the LDA, which is much, much faster and scales very well to large data sets. And there's also a number of features that are new in PySystopic, as you see in the table. Then in the second step, we take those co-accessible regions and do the motif discovery. This for us is quite central, and we put a lot of time into this step by curating a very large collection of position weight matrices using all these databases that you see there, over 30,000 position weight matrices. And then we clustered all those motifs. Of course, we have not even 2,000 transcription factors in the human genome, so 30,000 motifs. There's going to be many motifs per transcription factor, right? So by clustering, first using a Leiden clustering and then a hierarchical clustering on each of the clusters, we collected different motifs per transcription factor. Then we found out that, in fact, maintaining all this diversity, so maintaining the different motifs per transcription factor is actually beneficial, rather than creating an archetype motif or a consensus motif for each transcription factor. How do we maintain this variation? Basically by using a hidden Markov model, where we can implant the different motifs, the different position weight matrices of one transcription factor as hidden states, and then use the hidden Markov model to score a sequence. And then the path will go through the motif that is more probable. And multiple position weight matrices can be used. So on a large set of ChIP-seq data, we found indeed that keeping this variation is here, the red curve, gives you the highest prediction of the ChIP-seq peaks. And then this works better than Homer, probably because it has much more motifs, and also works better than using the archetype motif, which is there in blue. In lighter red. Yeah, this is the number of factors that we find on the first position. So there's 1,553 human transcription factors for which we have at least one motif now. So if you take a ChIP-seq data set, you know the factor, you do motif discovery, and you say, okay, which factor is on number 1? Then the one with the clustered motif is give you the highest number of TFs on the first position. And in the third step, now we have the topics, the regions, and the motifs. Then we're going to link it with transcription factors based on expression, link the regions to the genes, and create these networks that you can then visualize in SCOPE, in UCSC, and Cytoscape. And here's an example on PBMC with the transcription factors found per cell type. An example of a network here that has the factors, the regions, and the target genes, and then the tracks in the genome browser. After this validation, we set out to use Scenic Plus on two completely new model systems for the lab. The first one is the mouse liver, and the second one, the mouse and the human cortex. So the mouse liver, we performed single-cell attack, single-cell RNA, and single-cell multi-omic profiling of normal wild-type livers. And we also performed a resolve experiment with 100 genes, single-molecule fish. You can see that both in the RNA and the attack, these large group here are the hepatocytes, and they form this salami that represents different states in the liver. Basically, this reflects the zonation, where the hepatocytes have an altered state between around the central vein and the portal vein, so pericentral to periportal. It's also recapitulated here in the SM fish, from yellow to green to blue, with marker genes in the RNA-seq data, and from yellow to green to blue, also in the resolve data. So this is from the pericentral side to the periportal vein. Then Scenic Plus predicts transcription factors. There's a set of factors that are known hepatocyte transcription factors, CVP, HNF, FOX, HNF4, 1-cut, that are general for all the hepatocyte states, and some that we find that are specific for the periportal side and the pericentral side, so we can start to draw slightly different GRNs for the different states in the hepatocytes. We use this system mainly because it's a convenient system to also test enhancers in by massively parallel reporter assays, so we could generate a library of 12,000 attack peaks, clone them and test them in the liver. We found about 30% of these to show activity. Why is that interesting? Because now we can do transfer learning on our deep learning models, whereby first we train a model on the attack-seq, predicting the topics, for which we can use all the regions, and then train a model again, but now trying to predict the pericentral versus periportal state, as well as predicting active or not active. So we have accessibility, place, and activity. And with that, you have three models to now explain an enhancer. Why is it accessible in hepatocytes? Why is it accessible more in pericentral? And why is it active? And the second case study is, but I will skip this. You can see it in the paper, because I did want to, for the last two slides, to show two applications, if you want, how we can use these predicted Scenic Plus GRNs to, on the one hand, predict differentiation, and on the other hand, to predict the effect of perturbations. For differentiation, we found that there are transcription factors for which, along pseudotime in a trajectory, the TF expression precedes some of the target genes, so we can quantify this effect and actually draw this on the UMAP as an arrow that shows a differentiation strength. This way we find transcription factors like OLIG. This is oligodendrocyte differentiation in the mouse brain. OLIG there shows arrows in the precursor cells that keep it there, while SOX10 has very strong arrows that going from the precursor to the intermediate cell state. So we think this way we can prioritize factors that have certain effects in the trajectory. Stop, or one slide? Yeah. For the perturbation effect, we used profiling of different melanoma cell lines that are in different states. Here we use a network, so we have TF and target genes, so we can train a model, a random forest regression model, that predicts gene expression based on the TF inputs, and then remove a transcription factor, basically a knockout, predict gene expression again to generate the simulated expression matrix, which you can add to the UMAP, and then we have these arrows that predict where the cells would go if you knock down SOX10, and this is exactly what we have by the SOX10 knockdown experimentally, that these intermediate cell states go in the direction of the mesenchymal state. And we predict that ZEP1, if you knock it down in 57, would go back to the melanocytic state. With that, you can do over all factors and prioritize transcription factors with the strongest direction along a certain axis, like the first principal component. And as we find candidate factors that would change the states of melanoma cells. I will skip the conclusions and go directly to the acknowledgement. So I wanted to thank everybody in the lab, our collaborators, our funding, the organizers here, the chair, Julio. Thank you very much, everybody.
Thanks so much, Stein. So I think maybe Malte and then Fabian, or yeah.
Thanks, that was a really, really cool talk. I wonder if you can generate cell-type-specific gene expression and you can model GRNs, how far are you from inducing pathway activity in particular cell types that potentially induce measurable phenotype differences?
Induce what differences?
Measurable phenotype differences.
Yeah, the signaling part we haven't really worked on so much yet. So it's, like yesterday we saw in Julio where you combine different databases for signaling into this. For the measurable phenotype, I think the first we have gone here is the prediction of the perturbation and say, okay, this we observe a certain axis. You have to observe it first. Of course, that's quite essential. If you have never observed it in your single cell data, it's hard to predict where it goes. But if you have multiple states, and then say, okay, now I mutate this transcription factor in this state, and I want to find those factors that go mostly to that state, that's, I think, a measurable phenotype, because then if you can recapitulate this experimentally, you can actually knock it down and see if it indeed, the cells are going in this direction. But there's probably still a lot to do. We are just scratching the surface, I think, of the prediction of gene expression, changes. And this is completely inspired by Cell Oracle from Samantha Morris, who I think pioneered this idea of the linear regression of TFs and the perturbation. And then Scenic Plus just recapitulates that function from Cell Oracle. So shout out to that, it's really great, we love that.
You know, we're also interested in prediction of gene expression under perturbations in other settings. And I'm wondering how you deal with this sort of at the moment setup of yours that you have those regulatory networks, I think similar to Samantha as well, that are cell type specific, right? I mean, you saw in this, I think you call it salami in the liver, that, you know, there's obviously a continuum of things similar in development. Do you think you can sort of find maybe the dynamics of these networks, how they would rewire sort of? Is there like a master network that if you just put in the transcription factor activity that then sort of specializes the particular cell types? Or is there a sort of more complex process needed to put all of this together in sort of a unified single setup that all of the cells are using?
Yeah, it's a bit complicated. I think for the liver, on the one hand, the UMAP shows a nice continuum. On the other hand, if you, for example, derive some latent variables like topics and so on, you can actually boils down to 3 or 4 areas. Then you can still ask, you know, how artificial is that? This comes from the data. Yeah, it's much more defined. Also, the resolve data shows that if you plot your genes on the spatial data, actually, it's not so completely continuous. It's more like zones, right? So that's one thing. And the other thing about how then, was there some master network? I think that's the case. So there's a master control network, let's say, of the hepatocyte cell type. And then we found some Wnt-related, which is really pericentral, and some, TBX, but we don't know yet which pathway is there. It's like anti-Wnt related on the other side that influence this network, mainly by repression. So there's a Wnt activity. When Wnt activity is absent, there's a repression of the pericentral enhancers that we have predicted. We're currently still trying to validate this, but it's basically the influence of adding a transcription factor onto the network that's already operated by the cell type.
You have some additional controls, so to speak, that
yeah,
sort of make
inside some of these enhancers, right? So because the question was also, do you then have other enhancers around the genes that are controlled by this other thing? And so then you have some kind of a competition around the target gene, or the input goes inside the enhancers of the cell type. And both can still work. I think in the liver, it's more the latter.
Thanks.
Great. So thanks again, Stein, and yeah, we'll go to...
Thanks.
We have two short talks. The first one by Maryna Korshevniuk, there she is, who will talk about interindividual differences in the regulatory wiring of cells, provide novel perspective on disease.
Yes, I'm very excited to be here and present our recent project. And brief about myself, I'm a PhD student in Leuven group, and we mostly work on the understanding the interplay between genetics and also the cell type specific responses, and in general working intensively with single cell data. And indeed, today we'll try answer the question how the interindividual wiring of cells can gain us the new insights into the disease. But to start with this, I would like, oh, it doesn't work. It doesn't work for me. It works, okay, good. And to start with this talk, I would like to highlight the main question of the functional genomics is how exactly genes impact their traits in the population. And because most of these mechanisms remain unknown, we can consider it as a black box. And we have to, in order to tackle the disease better and to understand this disease progression, or as well as health state, we have to uncover this black box. With the recent advent of the single-cell technologies, it became possible to put the light on this, on part of this mechanism, because as all of us know, first of all, single cell gives a unique opportunity to go into the cell type specific resolution, but also it provides the multiple measurements of multiple cells from the same individuals, which is actually the very prospective basis for personalized medicine. And by interplaying and connecting the single cell profile as well as cell type specific data with genetics, we can also gain this predictable power into our research. So what do we mean by predicting what do we mean by matching genetics with single cell profile could be described as the EQTL effect or alternatively as the GeoAssess, but in general, let's zoom first to the EQTL effect. So what's going on the EQTL? You associate the level of gene expression and consider it as a trait with the particular genetic variance, which is a single nucleotide polymorphism or SNP. And by associating particularly these genetic variants with the level of gene expression, you can say that indeed we can predict this level of expression by knowing just the genetic variant. However, we all know that genes do not work individually, and genes have this particular interplay which lead us to the disease phenotype or other traits. That is why it is important to zoom into multiple genes and uncover this gene regulation and also gene interaction in cell type specific individualized manner. To address this problem, we developed a tool called co-EQTL mapping, where we map, instead of expression of genes derived from single-cell matrices, the co-expression of genes with a set of genetic variants. And this tool gives actually this opportunity to understand the regulation in genes and also zoom into cell type specific responses. So after this co-EQTL mapping, what do? We can zoom into the biological mechanism and among these identified gene pairs, find out the new transcription factors and other regulators which are relevant for our particular case. And as you can see on this slide, indeed we can find and associate known transcription factors to the E-genes, like genes related to EQTLs, and therefore better understand the biological mechanisms. And as a long-term perspective or long-term goal, we can further use it for patient stratification or in general prediction of level of expression and most important co-expression of genes. So now let's dive into the technical details, how we can do this with single cell data and what we actually did in our research. So we started with analyzing three studies, which could be split into 5 data sets based on the 10x version chemistry types. And as you can see here, so on this pie chart. So overall, we had a little bit less than 200 donors and 2 types of chemistries, which were treated separately. Between these 5 data sets, let's continue with number 5, just because it's more convenient. We found the similar abundancy pattern in terms of the cell types, and we identified the major cell types, and we're continuing on this higher resolution. Why it is so? Why cannot we zoom into the lower cell type resolution? Because in single cell EQTL analysis, you should have a lot of samples, and therefore, with a lower number of cells per lower resolution, it's not possible. So yeah, figuring out the number of cell types here, we decided to continue, and after that, we were checking out the concordance of the expression patterns among these cell types. What we identified that among different cell type specific matrices, the expression and consequently co-expression patterns were very concordant. As you can see here, average correlation of the between data set that we have was around 0.8, and this correlation was higher if the chemistry type was similar, as well as we had the high number of donors. We decided also to look into the bulk data and try to understand if we can utilize this bulk data in order to identify this co-expression relationship. And first of all, we started correlating these bulk expression profiles from fact-sorted cell type specific data with single cell profiles. And we ended up with this low correlation. So as you can see here, 0.4 or 0.5 is very weak correlation. That is, indicates us that maybe bulk is not the best thing to use for co-expression QTL analysis, like in general, matching the bulk and single cell profiles. What is even more interesting, that bulk data was even not concordant between each other. That is also an indication that, of course, we have concordant single cell data with a kind of big number of data sets for single cell, and it's probably better to continue like that. So with identified strategy, we were able to find 72 different SNPs associated with a little bit less than 1,000 of gene pairs, which were linked to, what is even more important, still replicated in bulk. It means that even when the differences of the sample size between bulk samples and single cell was in 15 times less, so we had like in 15 times more samples in bulk studies, all of the EQTLs found in single cell profiles were well replicated in bulk. It is reflected by this RB score. And even more, by this kind of analysis, we proved that a single cell has this higher sensitivity in terms of co-EQTL identification. Among all of these co-EQTL that we found in 6 major cell types, we found one EQTL popping up in every cell type. And this EQTL was RS1131. I will just say it like that because other name is quite long, which was linked to RPS26 gene. And looking into this SNP and gene pair, it doesn't seem very obvious how exactly this SNP could be linked to this ribosomal protein, and what is more important, could be described and linked to the autoimmune diseases. That is why we tried, by our strategy, we tried to explain how our ribosomal protein subunit 26, or like this part, like very obvious ribosomal gene, was linked to autoimmune disease. We tried to answer this question by our co-expression analysis. And actually, we found the suggestion how it could be that for some cell types and for this particular SNP and gene combination, it is a possibility to be linked to the autoimmune diseases. And let's zoom into this co-expression network that we identified. So what is in this slide? On this slide, you see the cell type specific gene pairs related to CD4 T cells and monocytes. And I would like to zoom into these 2 clusters on the bottom. So you see these gray gene pairs, which are common for both monocyte and CD4 T cells. And they are all, in terms of gene ontology enrichment analysis, they are all linked to the transcriptional and translational processes because they are linked to ribosomal, other ribosomal genes. But if we will zoom into another cluster, which is specific for CD4 T cells, we will see the high enrichment of lymphocyte activation, which is linked to, of course, inflammatory processes and therefore autoimmune diseases. So this is an awesome example how particular co-expression pattern discovery could provide the new insight and hypothesis that the gene and also SNP not linked directly to the autoimmune disease could be further explained and matched with these disease phenotypes. So what the strategy that we were using in discovering and explaining all the identified SNPs was the following, that first of all, we zoomed into the GWAS signals linked to these particular SNPs. After that, we zoom into the co-E genes, like gene pairs and co-express genes with this, which were linked to this SNP and gene pair. And consequently, we continued with the enrichment analysis of GWAS signals as well as gene ontology enrichment. And lastly, we zoom into the transcription factor enrichment and the transcription factor binding site analysis and identify potential upstream regulators of these co-expression activities. So one of the last bullet points that I would like to make in my presentation is that it's linked to one of the first slides that I presented, that as more samples, as more cells you have, as more precise you will get. It's also something that we were hearing yesterday on the presentation. And indeed, our research, as we were finding most of the EQTLs for the cell types that we had the most cells for, we showed also the clear linearity between the number of samples and number of cells and the number of consequently found co-expression QTLs. And we were lucky that recently we initiated a single-cell EQTL gene consortium where the main aim is to collect the data from more than 2,500 of donors and do the federated analysis of cell type-specific EQTLs. And now what we are working on is adjusting and adding to the strategy identification of co-expression QTLs. So with all of these robust co-EQTLs that we are going to identify, and we already identify in this proof of concept study, we are going to zoom into the personalized network, in particular by using the tool recently developed in our lab called Downstreamer, but also by recruiting the other multimodal data in order to understand this gene regulation on better resolution. So if you will have any questions or would like to continue discussion, I'm happy to answer on them here, but also I'm here around, so yeah, I will be happy to talk to you further. That's it.
Yeah, thanks a lot, Maryna. So we have time for 1 or 2 questions. If not, I'll start. So you found this association with autoimmune diseases, which I guess is a big umbrella of conditions, and okay, it makes sense they're all connected to T-cell. But so can you go more specific to a specific disease or?
Yeah, sure.
Or yeah.
Okay, so with this RS1131 SNP, the biggest enrichment and the main disease that we identified was type 1 diabetes. But overall, first of all, I'm on this particular EQTL, but also with other co-EQTLs that we found, they were linked to the asthma, multiple sclerosis, rheumatoid arthritis, and this kind of disease. So as I said, like we were first mapping them by the GWAS signals. We cannot go deeper so far, and we will probably continue with further analysis of these kind of phenotypes associated.
But if you would try to go like to one of them, you would not have enough statistical power, or? Like
for this kind of research, we might need a particular type of population where the genetics and single cell data is available. And this is, of course, the main struggle to find this population size as well as specific cohort. But as soon as we will have this data, we'll probably zoom into that.
Okay.
And in terms of this personalized network, we have the intent to match it with the medical records, or also disease history, but it's also just on the research.
Okay, great. I think Malte, one question. Yeah.
Sorry, thanks for your talk. It was really interesting. I'm wondering if you have the link, like if you have a link between SNPs and co-EQTLs, then you mentioned you kind of have target the genes kind of, you go to transcription factor expression as well. When do you have are you looking into the kind of mechanistic hypothesis from how you go from this SNP to the co-expression, or like, are you making that link through as well at the moment, or how are you?
Did I understand you correctly that first of all, we look into the co-expression of particular transcription factors to make this mechanistic interpretation?
I heard that you mentioned at the end briefly that you're also looking at inferring transcription factor expression as well. So are you making the mechanistic connection from end to end, basically?
This is one of our biological interpretation strategy. As soon as we found these gene pairs, we looked into that deeper. In particular, we looked if this, like the second gene or COE gene could be a transcription factor for this particular type of tissue or cell type, if the information available, or if the SNP located in this triplet of SNP E gene and COE gene could be located in the transcription factor binding site. And that's how we link it to the other transcription factors that we might found.
Thank you.
Yes.
Okay, great. Thanks so much, and we should move to the next talk. So the last talk of this session is by Florian Barkmann from ETH Zurich, who will talk about CanSig, discovering de novo shared transcriptional signatures in single-cell cancer cells.
Exactly. Thank you. As already mentioned, my name is Florian Barkmann. I'm a PhD student with Valentina Boeva at ETH Zurich, and we have developed a tool called CanSig for discovering shared transcriptional signatures in malignant cells. Before I start, maybe like quick show of hands, who here is working actually mostly or primarily on cancer? Oh, that's fewer people than I'd expected. Okay, but the first question I have is kind of probably for the people that mostly work on cancer. Can somebody basically tell me which of these cancer types has the highest recurrence rate? Okay, then maybe I just continue for the sake of time. So it's glioblastoma with basically above 90%. And these numbers all are really high. And this is, of course, a big problem, because we, of course, want to prevent people from getting cancer again, but also it really decreases the quality of life of patients because they always have this fear that the cancer comes back. And to kind of understand how we can help, we first have to think about a little bit about tumor heterogeneity. Because it's actually it's a really important parameter in recurrence of cancer. And the mechanism that we expect is that we have a very heterogeneous population. We expect this one to be more resistant to stress. So you can kind of see this in the graphic, where we have on the left-hand side a heterogeneous tumor. So we have a lot of like, whether in one state, all these like blue guys, and then we have the yellow guys representing a rare subtype. Then we apply chemotherapy, and what actually happens is this yellow guy survives, and then later on potentially does a state switch again, becomes proliferative, and then we actually have recurrence of the tumor. And we kind of want to do, hopefully prevent this from happening and identify these yellow guys to then actually target them with treatment. How do we want to do this? We want to identify gene signatures that then actually describes these yellow guys. So we want to take tumors from a lot of different patients, then hopefully find a latent space in which we can integrate them, where they cluster by their transcriptional state. And then from this transcriptional states, we actually then want to identify signatures that basically describe each of these states. And there are a lot of challenges associated with especially malignant data. Johanna Klughammer had already mentioned some of them. For example, we see that we don't see the subclustering by cell type in malignant data, but rather they cluster by patient ID or by patient. And this is due to a copy number variation, but also due to technical batch effect. And we also have a lot of other co-founding factors, depths of sequencing, and cell cycle. And these are all effects that we kind of need to get rid of to then identify these shared transcriptional states. And we hope that we can do this with our tool that's called CanSig. Okay, so this is basically the big overview over the method, and now we go into detail and we'll briefly run you through the entire pipeline. So for pre-processing, it's relatively standard. We take as input raw counts cell gene matrix with cell type annotations, and then we do standard quality control, so we remove low count cells and ensure that basically every barcode corresponds to an actual cell. We then use infer CNV pi, so there's a Python implementation of the infer CNV method to infer copy number variations of all the malignant cells, or of all the cells from the RNA sequence. And then we basically use the inferred copy numbers to also improve the split into malignant and non-malignant cells. So we make the assumption that all malignant cells actually show copy number variations. And then we end up with basically three matrices. One, the gene expression of the malignant cells, the copy number variations, also with annotated subclones for the malignant cells, and then the non-malignant cells. We then turn to integration, and here we actually want to leverage the inferred copy number variations that drives the differences between patients, as well as the cells, the non-malignant cells from the tumor microenvironment that are equally affected by the batch effect. And we do this by having two models. In total 3 models, but we first want to learn representations of the CNVs and then also of the batch effect. For the CNVs, we take the CNV profiles that we have inferred before. We take a variational autoencoder, embed them in a lower dimensional space. And anybody who has ever worked with CNV data inferred on RNA data knows it's a really noisy measurement. So kind of to combat the noise, what we do, we take the mean representation in the latent space per subclone. And this really stabilizes then basically for each cell this representation. For the batch effect, we make the assumption that basically every cell in the batch is equally affected by the batch effect, by the technical batch effect. So we can basically learn this from the non-malignant cells that we would otherwise not use for our analysis. And again, we do this by using SCVI, basically. We embed them in a lower dimensional latent space, but here we don't condition on batch, but we rather condition on cell type. So we kind of want to do the opposite of batch integration. We want to find a latent space where we really see clear clusters of the batches. And then again, to basically link A to make this representation more stable, but also now we have a representation that is per batch of non-malignant cells. It's not very useful for the non-malignant cells. We again take the mean representation in the latent space per batch, and then basically associate that with the malignant cells, where we also, of course, have the batch annotation. Then we go to a final autoencoder, where we basically condition on these learned representations, so our CNV representation as well as our batch representations. And this then allows us to actually control for these effects. And we also add additional covariates such as cell cycle scores or percentage counts of mitochondrial genes or log counts to get rid of these additional covariates. Okay. Then we run relatively simple post-processing on it. So we have a latent space. We run the third clustering method on it. We typically use Leiden clustering. Then for each of the clusters, we run differential expression analysis, and this then results in a list of genes. What we want to do, we want this to be stable across the choices of hyperparameters, and also it should be reproducible. So we kind of encourage users to do this multiple times. And everything that you see now, you can basically do from the command line without any computational experience. So we encourage you to basically run multiple steps of integration across different hyperparameters. And we also then run multiple post-processing steps, again, with multiple hyperparameters. So different number of clusters, different random seeds, potentially different algorithms. Which then results in a really large number of signatures. So every time we run post-processing, we basically get a ranking of genes for each cluster. And so now we basically end up with 400 signatures, so basically 400 rankings of genes. This is, of course, not very useful to the end user. We don't want to provide you with 400 signatures and you pick the best one for your publication, but rather we want to actually summarize them into meta-signatures, so signatures of signatures. And we do this by using clustering. So first we use weighted Spearman rank correlation to define a distance matrix on the space of signatures. And we use a weighted version of it because we really care about the top genes in our signatures. We won't use a tail end to define potential transcriptional states, but we really want to use the top of the list. And therefore it's weighted, and we give basically 80% of the weight to the first 1,000 genes. And then we use hierarchical clustering method to actually find these meta-signatures. So basically now we have a cluster of signatures, and then we take the mean rank for all these signatures. That's basically then the ranking that we associate with that meta-signature. And then you can do things like GSEAE, so gene set enrichment analysis, to identify pathways with these meta-signatures. Yeah, and we basically applied this entire approach as a validation to a known data set. So this is a glioblastoma data set. And in NAFTA, they identified basically 4 stable states that you can see here on the left-hand and the right-hand side, left-hand side. And the coordinates are not UMAP, as you would probably expect in a single-cell talk, but they found a way to basically turn scores into coordinates. And basically each quadrant of this plot represents one stable state. And we actually, with our method, we can actually rediscover 3 out of these 4 states. On the top left-hand, so you see our meta-signature that actually corresponds then to AC-like. And then if you go down, basically we find the other 3, except for meta-signature 2. This doesn't depend, it doesn't correspond to the signature that they identified. But there I have to say we did it on a different data set because they only published TPMs and our method actually needs raw counts as input. So we used a different glioblastoma data set. And this is also potentially one of the reasons why we are not able to identify all of their states. Then on the other side, you can see then the correlation matrix where we also can see that our meta-signatures very well actually correlate with their signatures. Okay, wrapping up. So to kind of like give you a good overview, what can you actually do? With CanSig, you can actually detect chat transcriptional signatures across patients in a stable and reproducible manner. So this is really important to us. We want this to be stable, of course. We have more validation on this on my poster, but also we want this to be reproducible. There's been a lot of work in this field, but people tend to write like their own 4,000 lines of R code, and nobody's ever going to be able or willing to redo the analysis. So we really hope that we provide a really easy tool to redo these analyses. Then you can also link these shared transcriptional states to biological processes, to GCAE, and we also found a way to potentially link them to copy number variations, but this is to be something you have to take this with a grain of salt because inferring copy number variations on RNA data is inherently noisy, and then this hopefully helps you to quantify interpretation transcription heterogeneity, which you can then target for personalized medicine and other things. I already want to thank my lab for a great collaboration. You can see the 3 people that were mostly involved in the project here as well. And I would really also thank the open source community. We heavily rely on tools built by other people. So there's ScanPy, there's SCVI tools, which we use quite a bit. And then there's also interesting VPy by guys from Innsbruck, from ICIB labs. Yeah. So now I have some time left for questions. You can always come find me at poster 14 if you have additional questions. We have use cases for people that are more interested in the biology that can kind of like showcase what you can actually do with the tool. We have written our own benchmark to try to understand how we can actually do data set integration of malignant cells and try to simulate data there. So if you're interested in that, also come find me. And we have additional stability analysis and things like that there. Also, there's a preprint out. You can find it there on the link. It's also on the poster. And there's, of course, link to the method with well-documented code and also like a page for documentation. So if you want to use it, let me know. OK, I think we have some time left.
Yeah, thanks, Florian. Indeed, we have time for 1 or 2 questions. There's one at the back.
I was wondering, so you mentioned that you do kind of combine results of different hyperparameters that you have run with. I'm wondering why you think that's, if you think there's no problems, for example, if one hyperparameter overcorrected in your integration and then you lose biological signal, wouldn't that also mean that you lose the biological signal from your final results when you make sure that this is consistent across all the different settings? I'm wondering what's your rationale for not trying to find the best setting for the hyperparameters rather than just combining across all of them?
That is a really good question. I think the first kind of like answer to this, it's really hard to know what is best. Like we can't really, in a latent space where you don't have cell type annotations, it's really difficult to know what best means. And to the second point of your, like, when we overcorrect, what we kind of make sure is that actually each of the signatures or each of the meta signatures contains signatures from all integration runs. So in that way, we kind of make sure that the signatures are not purely driven by that you found a latent space that is very easy to cluster in a certain way. And so kind of we flag runs when you have only, if you find a meta signature that only consists of 50% of integration runs, we flag that run for the user and tell them that's potentially driven by hyperparameter choices and not actually by the underlying biology. Hope that answers the question.
Good. Maybe Omer, yeah.
Nice talk. So the second point you made at the end about linking CNVs to shared transcriptional signatures. So this might be limited by available GBM data sets, but you know, GBMs show these really prominent chromosome 7 and 10 aberrations. There's things like EGFR mutations, on amplifications in chromosome 7. Have you been able to link any of your signatures to these well-known hallmark CNAs?
We actually haven't done it on, we haven't done it yet. We have mostly worked on different data sets. But really good idea.
OK, so we are finishing this session. So